Oh my, hi to my. Welcome to the third Raising the Bar Home Edition talk. Thanks for joining us tonight. I'm Fiona Scanlon from the Alumni Relations team at the University of Auckland. Taking research and education out of the lecture theatre and into city bars is what Raising the Bar is all about. Raising the Bar Home Edition takes it a step further, bringing the latest scientific research from the university right into your home. We kicked off the series 2 weeks ago with Professor Andrew Schelling, who talked about genetics, and last week, Associate Professor Matheson Russell educated us about democracy. To watch their talks, you'll find the recordings on our alumni website. In a few moments, we'll hear from Dr. Helen Murray, a research fellow at the Centre for Brain Research at the University of Auckland. Helen is also the captain of the New Zealand women's ice hockey team, and will give us an insight into what we know about the brains of contact sport athletes and the potential link to dementia. Our MC tonight is Helen's colleague, Dr. Victor Derricks, whose research team is investigating the early changes in Parkinson's disease. Victor will introduce Helen and will keep track of your questions to be answered later. So make yourself comfortable with a drink of your choice and enjoy the show. Over to you, Victor.
Thanks for that, Fiona. It's my absolute pleasure to introduce to you tonight Dr. Helen Murray. I had a quick look and went back into my memory, and it seems that Helen and I are going back over 10 years now. So the first time I met Helen was in the lab, where she started as a newly postgrad, trying to test different things in the lab, not really knowing what to do. The year later, she started her PhD in our group, and I thought at the time it would be a good idea to bring her closer into the rest of the group. So I suggested that she move next to me. Little did I know that Helen doesn't hold back, and she started asking questions on a daily basis, and it ended up being over 20 to 30, even 40 questions a day, which is, as a scientist, a really good thing. And over time, these questions got harder and harder, and I gave more difficult answers. For some people, this might be too much, but I knew Helen could handle it, because at one time, after a year after or 2 years after, she invited us to her different life, which I'm sure she will talk about today. Little were we know, where we were in the lab, that Helen had an alternative life playing ice hockey. So we went to this ice hockey game. It was my first game ever. And I remember she was playing the championships against Poland. And I was amazed by how powerful and intense this game was, until at one point even the glass got broken from one of the impacts. But the girls, they didn't mind at all. They kept on playing. And that's how Helen has been going on in the lab. She's taken a few punches, figuratively, but she's come and did an amazing PhD. I no longer call her my student, but I call her my colleague, and I'm happy to hand over tonight to her, and I'm keen to hear her story, how she tells it to you. Helen, over to you.
All right. Thank you, Victor, for sort of sharing a bit of our history of how we got to know each other. Yeah, it is an absolute pleasure tonight to be speaking at Raising the Bar. It's a series I've really admired over the years, and yeah, quite special to be able to sort of share, as Victor mentioned, sort of these two parts of my life. They're really big passions of mine, and they've kind of come together recently to form a research program, which is really just super exciting, and I'm really excited to share that with you. So I study a brain disease that affects people who have had repeated head knocks, and yet I also play a contact sport. So I actually started playing inline hockey when I was about 10, and inline hockey is like a version of ice hockey on rollerblades. It's very similar. And then I moved into ice hockey when I got a bit older, and I started playing for the New Zealand junior women New Zealand inline team when I was about 10, and then moved into the senior women team and then the Ice Ferns in 2013. So I've been playing hockey for a really long time. It's a huge part of my life, just as much as science is. And so while those two things might seem somewhat at odds, someone who studies the brain and yet also plays a very physical sport, I think they've been very complementary. And the skills that I've learned and developed as an athlete has also shaped me as a scientist. Both science and ice hockey require a lot of similar traits, a lot of teamwork, a lot of perseverance, a lot of hard work, and a lot of fundraising. So with all of this, I guess maybe it's no surprise that my research has sort of transitioned to focus on a disease that now we know affects people who have had a lot of head knocks over a long period of time. And while there is a lot of work being done on this question of, what happens in the brain when you get a head knock at the time, I actually study a disease that is occurring 10 to 20 years after these head injuries have occurred. So this disease is called chronic traumatic encephalopathy, which is a huge mouthful. So we're going to call it CTE tonight. And it's a progressive brain disease that affects people who have had these repeated head knocks over a long period of time. And many athletes aren't aware that these head injuries could increase their risk of developing a brain disease. But I think more and more in the media, we've been seeing people coming forward and talking about their experience playing professional sport and where their head injury has sort of meant that they're now sort of suffering from symptoms later on. And I'm sure there are many people around the world and in New Zealand who aren't realizing that actually something is happening in their brain. And this is a disease that we don't really know much about. Most of the research on CTE has been done in the last 10 years. So it's very recent, and we're still really in the early stages of figuring it out. And the first time I actually heard about it was also quite recently. So when I saw the movie Concussion, which was starring Will Smith in about, I think it was 2015. And by that time I was halfway through my PhD already. So it's been kind of a journey for me to kind of come into this research. And throughout this talk, I'm going to take you through that story of how my expertise in studying brain tissue has kind of come together with my life as a hockey player to kind of shape this research program. And by the end of it, I hope you'll know more about this disease and more about the research that we're doing to understand CTE. So first of all, we'll just set the scene a little bit. So throughout this talk, you'll hear me compare Alzheimer's disease and CTE. And these are different degenerative brain diseases that cause dementia. And this term dementia is an umbrella term, essentially, for a range of cognitive symptoms that can affect a person's life, such as memory loss, impairments in perception, reasoning, attention, language. And people with dementia can also experience psychological symptoms like agitation and depression and aggression and apathy. So Alzheimer's disease and CTE are types of dementia, and there are many others, like vascular dementia, Lewy body dementia, frontotemporal dementia, and of course, chronic traumatic encephalopathy, or CTE. And so what we find with these diseases is that the symptoms that people exhibit can actually look quite similar. And what really sort of differs is which symptoms sort of start first. And that can make it really difficult to diagnose what type of dementia someone might have. And so the only way to definitively make a diagnosis of what type of dementia they have is to look at the brain after someone has passed away. But if the symptoms are really similar, then people kind of ask, why does it matter what type of dementia someone has? Well, when we look at the brain, we see that there are very specific microscopic changes that happen in each of these diseases, and we call these pathologies. And by understanding the similarities and the differences between the pathologies in each disease or each type of dementia, we can build a picture of how that disease has developed. And that is really crucial for us to find ways to prevent or to delay or to treat disease. And so that's where my story starts. During my PhD, I studied Alzheimer's disease with Professor Maurice Curtis and Sir Richard Fall at Centre for Brain Research. And I worked with human tissue that was donated to the Neurological Foundation Human Brain Bank, which is based at the Faculty of Medical and Health Sciences and the Centre for Brain Research. And so throughout my PhD, as Victor mentioned, I sat next to him for a long time. I learned how to preserve brain tissue and to prepare these ultra-thin slices of tissue for my studies, and how to apply very specific fluorescent labels that would bind to only one part of the tissue that I wanted to study. So that could be something like a blood vessel or a certain type of brain cell or one of these pathologies that we wanted to understand. And throughout that process, I learnt how to study different processes within the brain tissue. I learnt how to take microscope images of the labeling that I was doing like this. And so these images allow us to kind of look at what's happening in the brain on a microscopic level. And throughout the course of my PhD, I was studying Alzheimer's disease. And the main kind of character in the project that I was looking at is this protein called tau. And so tau is a protein that has a normal function inside of our cells. It has a really important function to keep brain cells sort of structurally stable. And in Alzheimer's disease, they become, the tau proteins become altered and they start to clump together. And that causes them to form these dense structures that we call tangles. And so tangle pathology is not just a feature of Alzheimer's disease. And I guess, spoiler alert here, CTE is one of the diseases where we see this tau protein start to form. And the difference between these diseases is kind of where the tau starts to accumulate in the brain and how it moves around the brain as the disease progresses. But I'll go into that a little bit more soon. So I had learned a lot about tau and Alzheimer's disease by the end of my PhD. And I wanted to continue my research and I wanted to find out what was happening at the earliest stages of this disease so that we could sort of find out what are the things that we could intervene and what changes are happening that we could get to first and prevent the disease from even starting. That's kind of the real dream, is to make it so that no one even gets Alzheimer's disease. And so to do this, I developed a collaborative project with across 2 places. So I was fortunate enough to receive a philanthropic fellowship from the Health Ed Trust. And that was really, really amazing because it meant that I could kind of craft a kind of project which is a little bit different from how most fellowships might work, where you would be based in one place. So instead of just being based at the University of Auckland, I also traveled back and forward to the National Institutes of Health in Washington, DC. And that was an incredible experience because it's a whole new way of doing research and they have amazing resources over there. And by going back and forth between the 2 places, I was able to learn how to use sort of new equipment and new technologies and then bring that knowledge back to New Zealand sort of straight away in a really interactive sort of way. So that was really exciting. And during that time, I was studying a part of the brain that controls the sense of smell. And so I was looking at a whole range of things, including blood vessels and inflammation. These are processes that can happen in the brain that we think might be some of the earliest processes that start to occur when the brain is under stress and is damaged. So the blood vessels can become sort of broken and leaky. And it starts to trigger this cascade of inflammation where immune cells start to come in and sort of try and fix everything, but they actually kind of end up making it worse. So I was studying all of this in the area of the brain that controls the sense of smell. And the reason for that is because the sense of smell is one of the first symptoms that occurs in diseases like Alzheimer's disease and Parkinson's disease. And so by studying this area of the brain, we were trying to get an idea of what are the first things that go wrong in disease. And this is a really important question, because if we had early warning signs that could tell us when something is about to go wrong or is starting to go wrong in the brain, then we can do something about it before we start to lose all those really precious brain cells. And so while I was working on this loss of sense of smell kind of issue, I was reading about things that cause risk factors in disease. And one of the things that kept coming up was traumatic brain injury. And so it turns out that even after a mild brain injury, the brain cells themselves can become really stressed and the blood vessels can become leaky and we can start to see inflammation. And these were processes that I was already studying. And so it was kind of sitting in my mind that these were the things that were happening in brain injury that could also trigger dementia. And by this time, I was also, I was still playing ice hockey. So I started playing for the Ice Ferns in 2013, which is the same year I started my PhD. And I first captained the team in 2016. And throughout my PhD, I've been going around the world to compete in world champs. And during this time where I was doing a fellowship between America and New Zealand, I was also traveling around to play in world champs for hockey. So there was quite a lot of movement going on where I was, training, playing hockey, and then going to America and then coming back to New Zealand. And I think throughout that time, I became more and more aware of the head injury that was happening in my sport and in my team. More and more of my teammates were affected by concussions, and several had been forced to stop playing hockey entirely because of the long-term effects that they were experiencing. And that was really devastating because, you know, witnessing these really amazing, dedicated women, talented hockey players who are my really good friends, have to call time on the game that they really love and put pause on their study and their careers because of the symptoms that they're experiencing. Yeah, it's really hard to kind of watch. And being someone who studies brain diseases and has been reading about head injury and the risk factors for dementia, you know, I was starting to wonder, you know, what's happening in my brain right now? You know, where am I going to be and where are my teammates going to be in the future? And this is something I should be kind of worried about. And so I guess with any good story, there's always like an element of serendipity, and there is certainly a bit with mine. So during one of these trips where I was overseas and then kind of back in America, and I came back to New Zealand, in December 2019, I was back in New Zealand to do some work on the human brain tissue that was here. And the day that I arrived back in the country, Sir Richard Ford was hosting an event to launch a sports brain bank initiative, which meant that we were going to start receiving brain donations from people who had been athletes. And at this launch event, we were introduced to this disease called CTE. And it affects people who've had these repeated hits to the head over a long period of time. And we heard from a range of different people. So there was Sir Brian Williams, who talked about his head injuries from his rugby career and how that's affected him. And we heard from neurologists who have been seeing people in their clinic who have been athletes and experiencing symptoms of dementia. And I guess what was striking is they were showing symptoms in their 40s and 50s, which is a lot earlier than what we see with Alzheimer's, where people usually start to present with symptoms in their sort of late 60s. And then we heard about brain banking initiatives from around the world. So Associate Professor Michael Buckland is the director of the Australian Sports Brain Bank, and they had started a few years before ours, and they have since shown CTE. They found CTE pathology in the brain of people who have played rugby, and some of which were amateur players. Not all of them were professional football players or rugby players, sorry. So it really kind of highlights that this isn't just a disease of professional sports people. This is something that is affecting amateur players as well. And we also heard from Dr. Chris Nowinski, who is a former American football athlete and a WWE wrestler turned scientist who founded an organization called the Concussion Legacy Foundation. And he is championing this global sports brain banking effort. So he's trying to sort of get together these brain banks from around the world to start collecting brain tissue from people who have played these sports and experiencing dementia to see if we can learn more about CTE. Because if you want to understand how a disease works, the best place to start is actually by studying the people who have had that disease. So throughout this launch, we heard a lot about the brain changes that were happening in CTE. And most of the research is on these tau tangles. So we see them in CTE in a different area of the brain. So in Alzheimer's disease, we see them in this area of the brain that controls memory. That's where they start to form first. But in CTE, we start to see the tau tangles forming in the deep valleys between the folds in the brain. And more specifically still, they actually form around blood vessels. And so this pathology actually makes a lot of sense. So when you get a head impact, a knock, not necessarily a concussion, but just any kind of knock to the head, you get these forces that are concentrated around the areas where the structure of the tissue is sort of less consistent, which is things like around blood vessels and in these folds in the brain. And so these tau tangles are actually forming in the areas of the brain where the structure of the tissue is less consistent and in areas that we know are sort of experiencing the most stress. So yeah, eventually these tangles, they start off in these valleys, and then they start to move and grow and end up in other areas of the brain, including the area that involves memory and also other parts of the brain as well. And so this is quite important because it's very different to what we see in Alzheimer's disease. So in both diseases, you see sort of the tau tangles start to form in a certain place, and then they start to grow and move in other places. But in the later stages of the disease, and Alzheimer's disease can be classified according to 6 different stages, and CTE can be classified based on 4 different stages. But in these more severe advanced stages of these diseases, the brain tissue can look quite similar when you're just looking at the tau. And that can mean it can be quite tricky to diagnose CTE at these advanced stages, because we can't always obviously tell it apart from Alzheimer's disease. And that's just one of the issues that we need to address. We also don't know much about CTE other than these tangles. We don't know anything about what's going on with the blood vessels. We don't know what's happening to the brain cells, whether there's inflammation, and what's happening to trigger all of this. You know, what is the driver from the head injury to this tau forming and causing damage, and how is this similar or different to Alzheimer's? And so in order to diagnose CTE in living people, we really need to know what's happening at the start of the disease. We want to know what is the tipping point that determines when this disease starts to develop. What is the point when we would know that something is going wrong? There's no magic number of hits that will determine whether someone will get CTE, and not everyone who plays a contact sport will get CTE. So why is it that some people do? And for the people who do, you know, how do we modify or delay or stop this disease? And the first step to answering any of these questions is to figure out what's actually happening in the brain. And so I guess during this whole Sports Brain Bank Initiative launch, I had this kind of light bulb moment, because I realized this is a disease I really need to be working on. You know, this could be me, this could be my teammates. And everything I've been doing in my career so far had actually given me a really, really good foundation to contribute to this field. I knew quite a lot about tau and human brain anatomy, and I've been working on a project that explores early changes in these diseases. And probably most importantly, while I was in America, I was working on a new tool to study brain tissue, which could really accelerate the CTE research. And so when we study brain tissue, we apply 3 or 4 of these sort of fluorescent tags to see parts of the brain tissue. And that number, sort of 3 or 4, is because the microscopes that we use can only separate kind of 3 or 4 of these colors when we are taking the image. And because the brain is so incredibly complex, we often have to kind of mix and match these different tags and different combinations using different slices of brain tissue, so that we can see all the different things that we would need to see in order to answer a question that we wanted to know about the brain structure. But what we did in America at the NIH was design a way to label the brain tissue with more than 100 different tags, all on one slice of brain tissue. And that just took some relatively simple adjustments to a microscope and a little bit of changes to how we actually do the brain tissue labeling. And so essentially how this works is we make these adjustments to the microscope, which means we can label 10 markers on the tissue at a time. And then we take that image and then we remove all of those labels and we add another 10. And so by doing that, what we do is we build up this library of images of all the different tags on a single piece of brain tissue. And now we have these incredible images which allow us to see all of the complex and fine detail that we would need to see in the brain tissue. So rather than just seeing one marker of a blood vessel, I can see all the different layers of that blood vessel and to see, is there a specific part of the blood vessel that has changed or altered? I can look at different parts of a brain cell and see if there's a certain part that's changed or altered. And that is just incredible, because we get 20 times, at least 20 times more information from a single piece of brain tissue than we normally would get. And that doesn't only mean that we can ask better questions about what's happening in the tissue, but it also means we can really maximize the amount of information that we get from a single piece of tissue, which is such a really precious resource that we want to make the most of. So I guess what I find so exciting and incredible about this multiplexing tool is that we can use it to acquire a lot of information about the brain structure and CTE, which is what we really need to know in this field right now, is what is actually changing in the brain. We have so much to learn. And so rather than painstakingly labeling a couple of markers at a time, we can generate these amazing image libraries that allow us to see all this complex detail. And I don't think these data sets will just help us answer questions about CTE. I think it's also going to help us understand other diseases like Alzheimer's disease better as well. And so that is the approach I'm taking with my research. I'm using this labeling to explore questions like what is happening to the blood vessels, what types of brain cells are affected by that tau pathology, the tau tangles. Is it just the location of the tau that's different, or is there a sort of a different rate at which it moves and changes around the brain? Or is there a completely different process going on? What's happening with inflammation? There's just so many things that we want to learn. But of course, all of this research is dependent on us having brain tissue to study. So since our Sports Brain Bank initiative launched in 2019, we have had lots of people, amazing people, register to donate their brain to our research after they pass away. And it's an incredible gift, a really incredible gift from a family. And it's something that we absolutely cherish. And the families they're on this research journey with us. And everything that we learn from the brain, we pass on to them, because it's so important for the family to know, if their loved one had CTE, if they had something else. And that's something that we really want to be able to give back as well. So we're actively accepting brain donations and registrations to donate from people who have had a history of repeated head injury. But we also need to study brains from people with Alzheimer's disease and people who are sort of, cognitively normal, so that we can compare and actually understand what is different in CTE compared to these other forms of dementia or just from normal aging. And so if anyone is sort of looking for information about brain donation, I'll put a website, and then you can go to brainbank.ac.nz to get more information about that process. And yeah, for now, we are yet to receive a CTE brain to study in New Zealand. So we're working with the Australian Sports Brain Bank and some overseas collaborators to get this research going. And we've recently had an amazing philanthropic donation to get a PhD student on board, and she's just started her project, which is going to be looking at this multiplexing technology and CTE. So it's really exciting. Yeah, we really can't wait to get into it and see what we can contribute to this field. And yeah, so I guess, what I've learned in this career so far and from all of this research is that science is like sport in many ways. You need a really amazing team of people with different expertise and perspectives to come together to take on a huge challenge like understanding CTE. And I feel very privileged that I get to work with so many talented people. Not just scientists, but the families and the people who are out there living with the aftermath of head injuries and getting out there and telling their story. And yeah, I think together we can make a big difference by understanding the link between head injury and dementia. And I think we'll make some really good progress in understanding not only CTE, but other forms of dementia as well. And now I guess, okay, I'm a hockey player, right? So I have to quote Wayne Gretzky to finish. My favorite quote from him is, "A good hockey player plays to where the puck is, and a great hockey player plays to where the puck is going to be." So my message to athletes who play contact sports is to love your sport, because I love mine. And also remember the long game. Play to where you're going to be. One day when we hang up our boots, we still want to have a relatively healthy brain so that we can enjoy everything that life has to offer. And, the brain is the most incredible thing that you own. It's really, really quite amazing that everything about you is held in this one soft little organ in your head. And it really doesn't like being bumped around. So we need to look after it. But just know that if yours has been bumped around, we are doing this research and we are doing our best to get these answers. And New Zealand, I think, has a lot to contribute to the space with our great love of contact sport. So I hope I can use my sport background and my neuroscience expertise to get some answers. And with that, I'm going to hand back to Victor.
Thank you, Helen. That was a great introduction to your work. Just for everybody, some housekeeping. If you want to ask a question, please fill in your question in the Q&A section. Other people can vote the questions they like to ask Helen as well, so that we can ask the question that is most relevant for everyone. So you have time to do that now. In the meantime, I will ask the first question. So Helen, you talked about brain donations. So people that are wanting to help your research, how can they do it? And do the brains that get donated from you, do they need to come from like professional athletes from like heavy contact sports, or can it be anybody else?
Yeah, that's a really great question. So there's we know that the biggest risk factor for CTE is exposure to repeated head injury, and it isn't necessarily from hard knocks or having lots of actual concussions. It's just the exposure to head bumps. So you might not have had injuries that have caused symptoms like concussion symptoms, but you could have had lots of bumps across a really long period of time that can increase the risk. So it's not just professional sports people, it's also amateur athletes, people who have played a contact sport for a long time. Sometimes I think we don't even realize as athletes how many times we've actually kind of been bumped around, and really anyone that plays one of these contact sports and it has symptoms of dementia or symptoms of cognitive decline is kind of what we are looking for in terms of the brain donation program. The other sort of point you touched on is the brain donation itself. So there are sort of different processes to brain donation. It's not the same as body donation. We actually have to contact the people very quickly after they've passed away, the families, and we ideally need the brain about 24 hours, ideally after the person's passed away. So it's not something that you can leave to the last minute. If it's something that people want to consider, we really encourage them to get in touch with us, and we've got some questionnaires and sort of go through the process and talk to family about your wishes, so that we can really do the best research possible with a brain donation.
And with that, of course, you need people that are perfectly healthy as well, right?
Yeah, so one of the biggest things that we actually need is sort of a normal, healthy control to compare to, so that we know what is actually CTE, what is the disease, and what is sort of normal aging, so we can kind of tease those things apart. And brain donation is a really amazing gift. It's something that you aren't necessarily going to benefit from once you've gone, but future generations are going to get so much benefit from. And so I think it's a really, really incredible thing for people to consider. So I want to thank everyone already who has already registered. It is really incredible.
Great. So can you tell us a bit more about the multiplexing? What kind of changes do you need to make to get more color information? So what's the process to like get to those amazing images?
Yeah. So without sort of getting too technical about it, the way that these microscopes work is that they capture light in certain wavelengths. And so when we add one of these fluorescent tags to the tissue, those tags will emit light in a certain wavelength when they're excited. And so most microscopes will capture sort of only 3 or 4 different wavelengths of light. And so all we had to do was change our microscope in a way that it could capture sort of narrower bands of wavelengths of light. And in that way, with sort of these different filters, you're able to separate different colors or different spectrums of light. And that essentially is what allows us to get to this sort of more information on the tissue. And then I think the other thing that was quite game-changing is the ability to capture that image and then remove all of those labels off and kind of start again. So the tissue is still preserved. We don't lose any information off the tissue itself. It doesn't get too damaged. And we're able to then sort of layer the information iteratively over time, over a sort of series of rounds, in order to get these amazing images. And everything then gets aligned together, and then we can look at the whole image stack and compare things that we might have labeled in the first round to the 5th round, which is not something we've been able to do before.
It's almost like drawing a coloring book, where you used to be able to do 1 or 2 colors, and now you can do 100 colors.
Yeah, that's a great analogy, and it creates such beautiful images. And I think that it sort of reminds you of the night sky. It's just this incredible array of different things that you can see, and it just goes to show how incredible your brain is, because all of these things are happening there, and we haven't really been able to see them until now.
Yeah. Brain is absolutely an amazing thing we need to take care of. A question from one of the listeners, from Hillary. Is there a parallel between the loss of smell in early dementia in Alzheimer's or CTE and similar symptoms during COVID?
Yeah, this is a great question, and a bit of a segue actually, because while I was in America and I was studying the sense of smell, I also ended up working on a project that was looking at the sense of smell in COVID-19. So I guess when you're working on one brain region, it's sort of, when a pandemic hits, that's kind of what happens, right? So I guess the short answer is we aren't sure. It looks like in COVID-19, the sense of smell problems come from the cells in your nose that actually receive the odor information. And in Alzheimer's disease, we haven't quite figured out yet what is causing that sense of smell. So it could also be those cells in the nose that send information into the brain that is the problem in Alzheimer's, but we're still kind of trying to figure that out. What we did find in Alzheimer's is that we saw those tau tangles in that area of the brain that controls the sense of smell. So it was one of the first areas that was affected.
An interesting question, like you talk about rugby a lot. What are the highest, who's most at risk besides rugby?
Yeah, I think we think of rugby because, in New Zealand, that's one of the big sports that we love. And rugby players, are at risk of having a lot of head injuries over a long playing career. Overseas, though, the majority of the work that has been done on CTE has actually been done on American football players. So there is obviously quite a big American football sport in the States, and that's where people first started to realize that CTE was happening. But before that, this disease was actually found in boxers. So it was, back in the day, it was called dementia pugilistica, or punch drunk syndrome. And we now know that is essentially the same as CTE. The pathology we see in the brain is the same as what was described in boxers. So boxing is probably one of the sports that we know quite a lot about, also American football, but also sports that have been studied around the world include ice hockey, so it's me, and also football or soccer. So there has been some studies in the UK that have shown CTE and soccer players, and presumably because there's a lot of practice of hitting the ball with the head over a long period of time and long playing careers.
And so with these injuries, and I know we've been studying quite a bit of the professional athletes, but is there a danger when you start doing these sports earlier, like from a year early on as a kid? And does it matter how many times you practice? Is it the total amount, or is one big head knock worse than 10 small ones? Can you talk to that?
Yeah, that's a great question, and one that I think is quite commonly asked. We know that it's not necessarily the number of concussions that you have. So the number of concussions doesn't necessarily correlate with getting the CTE, because we know that quite a lot of people won't necessarily report their concussions as well. It's actually more about the duration of exposure to head injury. So how long someone's playing career has been, how long they've potentially been hitting their head for. So really one of the big drives at the moment is to try and reduce the amount of head injuries that children will be getting. So trying to start contact later, instead of, we don't need 5-year-olds bumping their heads. It's just not necessary. And if you can reduce contact until they're older, then we're not damaging young brains that are still developing, and we're also reducing the amount of head exposure by 10 years maybe, and that can make a really big difference in terms of the amount of trauma that the brain is experiencing.
And would that also work with, like, for instance, football players with when they do like headshots?
Yeah, exactly. It's more about, can we limit the ways that we train and play so that, limit the amount of head injury risk. Even those ones that don't necessarily cause a concussion, they're still dangerous. So are there ways that we can adjust our trainings and our games so that we can still play fast, exciting sport, but not take as much risk of these head injuries. And I think there's definitely ways that we can do that.
So your study is focusing on when you have the brain in the lab and you can image it. But Margaret was asking, is there a way to analyze CTE in people that are still alive? So you can actually diagnose and potentially then treat them.
Yeah, and that is really where the research needs to be. So the reason that we study the brain after the person has passed away is because we need to find markers in the brain to tell us what CTE is, so that we can try and look for it earlier. So in order to know whether someone has CTE, at the moment, we can only find that out from actually looking at the tau pathology. But we can't really see tau pathology with non-invasive methods like MRI or blood tests. It's not really conclusive for CTE. So right now, there's actually no definitive way to diagnose CTE in living people. But we hope that with our research and finding different signatures of what CTE looks like in the brain, we'll be able to combine this to find new ways to diagnose CTE in living people with more accuracy. And that will help us. It'll help us do a lot, really. It'll help us, as you say, try and find out when someone is at risk of sort of developing a degenerative disease. It'll allow us to find ways to intervene before things start to go downhill. Like, can we stop this cascade of damage before it turns into full-blown dementia? And to do this, what we really need to do is to follow people over a long period of time. So this is a kind of study that requires us to enroll people who might be at risk for developing CTE and monitor them over a really long period of time and see what's actually happening in their brain. What changes can we see on MRI or blood tests? And then one day when they do pass away, we'll look at their brain and see if they had CTE. And that's going to give so much context to what we actually see in the brain, because we know what it looked like early on. And that's what will allow us to figure out how to diagnose it.
You'll have a long time to
Yeah, it's a very long-term project, my whole life's work, I'm sure.
So for those people that they know they have repeated head injuries, is there already a way that they can change their behavior to preserve or improve their brain function, to actually do something now, even though they might not see any of the symptoms that might occur, but to work proactively?
Yeah, absolutely. I mean, we always say, I think at the Centre for Brain Research, and this is probably Sir Richard's quote more than anything, is what's good for your heart is good for your brain. So, exercising and staying fit and healthy and eating, good quality food and keeping your brain active is the best thing that you can do to prevent or delay or, any sort of cognitive decline. And I think that's probably one of the big take homes for me, is that knowing that you're at risk of developing something like CTE. So for someone like me, I love my sport and I want to keep playing it. And I know that I'm putting myself at risk, but by understanding that risk, I feel like I can make better decisions about how I look after myself, so I don't add any more risk factors on as I get older. So that's things like not drinking alcohol excessively and looking after my health and my diet and that kind of thing. So I think probably that's the biggest take home, is that if you think you might be at risk, to make decisions about your long-term brain health, that's going to make sure that you kind of reduce the risk as much as you can.
And is there an average age of diagnosis of CTE?
We're still really learning a lot about it, and as I said, it's kind of difficult because we don't have a conclusive way to diagnose it in living people. The best we can do is diagnose a probable CTE, and which will then be confirmed when the person passes away. So it is difficult, but we anecdotally and from sort of the limited studies we have, it does seem like people develop symptoms a lot earlier than other degenerative diseases like Alzheimer's, so sort of in their 50s or late 40s as opposed to 65 and over. And so I think that's also one of the things that make people not necessarily realize what's happening, because a lot of people think, oh, I'm too young to be getting dementia, or this can't be that because I'm not old. And it's true, they're not old, but we know that this is occurring a lot earlier.
Yeah. So we talked a little bit about the dangers of getting repeated head injuries when you're young, but I'm going to move to the other side of the spectrum, and we all know that when you age, you're more at risk of falls. Is there an increased risk for those people, the elderly, to get CTE, or is that will it take too long to develop the CTE in those people?
Yeah, that's a really good question as well. Actually, we know, I guess, from studying young people, athletes, that kind of thing, that exposure, the amount of time and the number of head injuries is the most important risk factor. But we know that as the brain ages, it does become sort of more vulnerable. It does become a little bit more prone to sort of developing pathologies or damage. And so falls later in life may potentially accelerate some of these changes when you get damage into those areas where you get stress and a head knock. Potentially that could accelerate some damage. So it's not something that we know yet. But I guess any head knock is probably bad from what we're finding.
Yeah. And this is a question from Sophie, which I find quite interesting. I never thought about this one. So really good question here. Are there any known genetic factors that contribute to people developing CTE? Because, yeah, so if you compare two people with each other, you'll give them the same amount of head knocks. One might develop, the other might. So are there any predisposing factors in addition to the genetics?
Yeah, I mean, another really, really good question, and something that I think we will explore as we start to receive brain donations and look at the genetics of the people that we study is there actually factors that will maybe predispose people to getting CTE? We know with degenerative diseases that, you know, it's sort of like a step ladder of risk factors. You know, you might be born with some genetic predispositions to a disease, and then over your lifespan, you might sort of take another step up the risk factor ladder if you add things like head injury or other sorts of things, like maybe, you know, living in an area with high pollution or something like that. So, you know, as we kind of take more steps up that risk factor ladder, we could potentially be getting closer to a disease. And so I guess if you start with a genetic risk factor, that would, potentially explain some of the reasons why some people can play a contact sport their whole life and not get CTE, and some people could. The short answer is we don't know. We don't know yet. We know that there are some genetic risk factors that can lead to sort of a predisposition for tangles to form, but it's not well studied in CTE at all, really.
And maybe a question to follow up on that, because I think it's quite fitting, because some of the you've highlighted a little bit before about the correlation between Alzheimer's and CTE. So are there similarities between your research on CTE and other neurodegenerative diseases such as Parkinson, Huntington's, motor neuron disease?
Yeah, we really want to understand these similarities and differences more by comparing the changes that we're seeing in the brain. There are similarities in the sense that we know inflammation is a feature of quite a few diseases. And we know that blood vessel damage can also be something that affects multiple diseases. But I guess the question is where in the disease process are these things occurring? And I think with CTE, we think they're occurring quite early. We think it could be one of the driving things that is leading to the progression of the disease. In terms of, like, you know, what are we learning about these diseases as well? I guess the complicating thing, which I hadn't mentioned until now, is it's not uncommon to see multiple pathologies that would be associated with different diseases in one person. So sometimes you can see classical CTE pathology, the tau in the deep valleys of the cortical folds, and you can also see features of that might be associated with Parkinson's disease or might be associated with motor neuron disease. It's quite common to see that. We don't really know what's going on there. We don't know if it's the CTE lesions start and then they can almost sort of predispose the brain to other pathologies starting to develop, or maybe it's associated with age. As people get older, they have a likelihood of developing multiple things. So there's a lot of complication there in terms of how these diseases are similar and different and how they can present in different people, which is a complicated answer, but basically to say there's a lot going on and we still need to figure that out as well.
Yeah, I think one of the issues will be because there's a long time between the symptoms and potentially the first incidents, right?
Yeah, that definitely makes it a bit more challenging as well. Yeah. And we typically study people at the end of their life where, they might have been receiving head injuries in their 20s and 30s and then developing this pathology since they were 50 and 60, and then we don't see the brain until sort of 20, 30 years after that. So it is a bit of a trying to figure out what's happened. Yeah, it's one of those sort of puzzles where you only see the aftermath and you're trying to figure out what happened at the start.
Yeah, just probably the same question, but a different flair to one that we've asked before. So if you have head injuries during childhood when your brain is still quite developing, or you have it later in life when you have like a fully formed brain, would you say that one is more dangerous to the other for developing CTE?
Oh, these are all really good questions. We know that during brain development, there is a lot going on in terms of how the brain cells are wiring and connecting up to each other. And that process is very dynamic. And it's really important that the brain cells are able to sort of find their connections to each other. And so I think, we don't have a lot of evidence on this because that would require us to be studying the brains of very young people. But we can sort of hypothesize that, any damage to the brain during that quite critical period where, brain cells are connecting and refining their connections isn't going to be a good thing. And so whether or not that predisposes people to getting CTE is, again, I feel like I'm saying this a lot. We don't know because we're still learning so much about this disease. But I think it's pretty clear to say that getting these head knocks early is not going to be a good thing in terms of brain development. I think that's a pretty pretty sure thing. So anything we can do to sort of limit head injuries at a young age, I think is always going to improve brain health over the long term.
And you've spoken a lot about sports, contact sports that are linked with CTE, but can you are there other risk factors? I'm thinking it's not just sports that is to blame. We've got car accidents as well. Does epilepsy and epileptic seizures form a risk as well?
Yes, you're right. So I have talked sort of exclusively, I guess, about sport, head injury in sport, but it's not just sport. It's anything that leads to repetitive head injury over a period of time. So that can be situations like domestic abuse, where people have had head injuries over a long period of time. It could be, epileptic seizures, if people have hit their head quite a lot over multiple times. There are lots of other reasons why people might have had a lot of head injuries, and those are all risk factors for CTE as well. It's not just sport.
Mm-hmm. Yeah, and as you said, of course, before, the brain is an amazing organ. So even if we get brain injuries, it doesn't stay still. It can repair itself, and that kind of like depends. So can you just talk a bit about that, how innate repair mechanisms are?
Yeah, so this, I didn't really mention it, but the idea of the brain being able to repair itself was something that I explored during my PhD. So that was one of the key things I was trying to understand, is whether or not the brain in Alzheimer's disease still has the capacity to sort of repair brain cells and repair itself. And the short answer is, the brain can grow new neurons, new brain cells in two particular areas of the brain. But in adult life, it doesn't do it at a rate that is fast enough to repair the amount of damage that's done in a degenerative disease. So I guess the short answer is the brain is incredibly plastic. It's incredibly adaptable, and it can compensate for a long time while brain cells are dying. And we don't typically see disease symptoms start to form until that kind of capacity to compensate is lost. Once too many brain cells have gone, the brain isn't able to compensate anymore, and that's when we start to see symptoms. And so while there is some capacity for the brain to compensate and to try and repair itself, it doesn't do it at a rate that is fast enough to really make a difference. And that, I think, is the main reason why we have such a focus on trying to prevent the disease, trying to delay it, trying to slow it down, because we know that once those brain cells are lost, it's very difficult for the brain to actually repair them.
Maybe a small side question that just popped up. Does alcohol exacerbate, like make the effects of head injuries worse?
Again, this is probably one of the hot topics at the moment in CTE research. Is a lot of people who develop CTE symptoms can go on to develop things like alcohol addiction and aggression and things that could potentially be associated with alcohol use. And it's very difficult to tease apart what is happening from the head knock and what is happening potentially from alcohol use. So yeah, it's very unclear at this stage.
Yep. And maybe one last question to finish off the discussion. Knowing what you know now about CTE and head knocks, would you go back and tell your younger self to still go and play hockey?
Yeah, I mean, this sport has given me so much. It's really sort of made me who I am today. And so I love sport, and I love being able to play sport. I think knowing earlier that head injury could give me, a higher risk of developing a brain disease later in life, I would have definitely taken a lot more caution, but also made different decisions about, when to go back and play. If I've had a knock, just to sort of stop and go, actually, I really want to play this game, but what's more important in the long term? And so I think, if anything, it has changed the way that I think about how I manage my health in terms of, if something happens during a game and how I deal with it. And I try and sort of remind my teammates as well, hockey, it's just a game, sport is just a game, and we have our whole lives ahead of us. So let's make decisions that benefit the rest of our lives.
Yeah, and that's something that's not just true for our brain, it's for everything that we do in life. We should take care of our brain and our body at all times. I think the questions keep on coming in. Unfortunately, I will have to finish the session. I would like to thank Dr. Helen Murray for her amazing talk, and the organizers for raising the bar. I think it's been a great session thus far. And for those who want, next week we have another session with the speaker Ellie Lowry, being emceed by Rebecca Jelly, and with a very interesting topic which has come up a little bit today, from the vineyard to the glass, what really goes into New Zealand wines. So if you want to sign up for that, you can do that on the website. The recording from today will also be made available on the website. I would like to thank you for attending this session and hope to see you back next week.